Welcome everyone once again to General Biology, Campbell Biology Textbook, Chapter 10, and this will deal with photosynthesis. The process that feeds the biosphere. Plants and other photosynthetic organisms contain organelles called chloroplasts. Photosynthesis is the process that converts solar energy into chemical energy with chloroplasts. Within chloroplasts, directly or indirectly, photosynthesis nourishes almost the entire living world. Autotrophs are self-feeders, in a sense, that sustain themselves without eating anything derived from other organisms. Autotrophs are the producers of the biosphere, producing organic molecules from carbon dioxide and other inorganic molecules. Almost all plants are photoautotrophs, using the energy of sunlight to make organic molecules. Other organisms also benefit from photosynthesis by eating those plants that do. Photosynthesis occurs in plants, algae, certain other unicellular eukaryotes, and some prokaryotes. One that will be often talked about, usually in general biology too, is types of bacteria like cyanobacteria. But the large diversity of life that can make use of photosynthesis spans many kingdoms. Purple sulfur bacteria. Heterotrophs obtain organic material from other organisms. Heterotrophs are the consumer of the biosphere. Some eat other living organisms. Others, called decomposers, consume dead organic material or feces. Almost all heterotrophs, including humans, depend on photoautotrophs for food and O2. Earth's supply of fossil fuels was formed from the remains of organisms that died hundreds of millions of years ago. Fossil fuels are being consumed faster than they are being replenished. Researchers are exploring methods of using the photosynthetic process to produce alternative fuels. Photosynthesis converts light energy into the chemical energy for food. Chloroplasts are structurally similar to and likely evolved from photosynthetic bacteria. The structural organization of these organelles allows for the chemical reaction of photosynthesis. Leaves are the major location of photosynthesis in plants. Chloroplasts are found mainly in cells of the mesophyll, the interior tissue of the leaf. Each mesophyll cell contains 30 to 40 chloroplasts. CO2 enters and O2 exits the leaf through microscopic pores called stomata. A chloroplast has an envelope of two membranes surrounding a dense fluid called the stroma. Thylakoids are connected sacs in the chloroplast that compose a third membrane system. Thylakoids may be stacked in columns called grana. Chlorophyll, the green pigment that gives leaves their color, resides in the thylakoid membranes. And here we're just looking at a variety breakdown of the different components that were just discussed inside a chloroplast. Usually most biology professors want you to know thylakoid as those stacks, and the stroma and granum, and as double enveloped membrane. And that this happens in the leaves. Tracking atoms through photosynthesis scientific inquiry. Photosynthesis is a complex series of reactions that can be summarized as the following equation. 6 molecules of CO2 react with 12 molecules of H2O in the presence of light energy, converting or rearranging the chemical bonds into a molecule of glucose, 6 O2s, and 6 molecules of water. That looks a bit suspicious. I believe that there is a... okay. For a net use of water being 6H2O being inputted, the overall chemical change during photosynthesis is the reverse of the one that occurs during cellular respiration. Chloroplasts split water into hydrogen and oxygen, incorporating the electrons of hydrogen into sugar molecules and releasing oxygen as a byproduct. Here we have our reactants. Our carbon goes into the future glucose molecule, with oxygen split between it and water, and similarly water donates its hydrogens, and oxygen becomes O2. Photosynthesis reverses the direction of electron flow compared to respiration. Photosynthesis is a redox process in which H2O is oxidized and CO2 is reduced. Photosynthesis is an endergonic process. The energy boost is provided by light. Photosynthesis consists of two different stages. One is the light reactions and the Calvin cycle. The light reactions involve the photo part, or the light-induced part, and then the Calvin cycle is the synthesis part. The light reactions, which happens in the thylakoid of chloroplasts, these reactions split water, they release O2, they reduce the electron acceptor NADP+ to NADPH, generate ATP from adenosine diphosphate by photophosphorylation, or light-induced phosphorylation. The Calvin cycle that occurs in the stroma forms sugars from carbon dioxide using that ATP and reduced NADPH. The Calvin cycle begins with carbon fixation, incorporating carbon dioxide into organic molecules. As you see here, the process involves the interaction of light energy reacting with, and this is more of a bioenergetics course that some may take future or in grad school. There's a series of molecules that light can interact with at different frequencies or wavelengths, and eventually be funneled down to have the energy reach where it's necessary to activate these photosynthetic reactions. Now what we're seeing is ADP plus becomes reduced, and ADP becomes phosphorylated to form ADPH and ATP respectively, which are used in the Calvin cycle to eventually restore ADP and ADP plus while incorporating carbon dioxide gas into glucose, sugar. So the inputs are light energy, water, and carbon dioxide for a net output of O2 during the light reactions and sugar during the Calvin reaction cycles. Chloroplasts are solar-powered chemical factories. Their thylakoid transforms light energy into chemical energy found in ATP and NADPH. Light is electromagnetic energy, also called electromagnetic radiation. Electromagnetic energy travels in rhythmic waves or oscillations. Wavelength is the distance between crests of these oscillations or between valleys of these oscillations of electromagnetic waves. A wavelength determines the type of electromagnetic energy. The electromagnetic spectrum is the entire range of electromagnetic energy or radiation. Visible light consists of wavelengths ranging from 380 to 750 nanometers, and those are the wavelengths of light that produce the colors that we visualize. Visible light also includes the wavelengths that drive photosynthesis to occur. Light also behaves as though it consists of discrete particles called photons. So what they're getting at there is to the wave-particle duality, which you will more likely cover in general chemistry or general physics. Gamma rays, X-rays, and UV light are forms of higher energy, higher frequency, lower wavelength light than visible light, and then infrared, microwave radiation, and radio waves are examples of lower energy, lower frequency, higher wavelength types of light beyond the visible light spectrum. Key here is that wavelength increases as energy decreases or as frequency decreases. And frequency and energy are proportional, but in-proportional to wavelength. Pigments are substances that absorb visible light. Different pigments absorb different wavelengths. Wavelengths that are not absorbed are reflected or transmitted. Therefore, when we look at leaves and we see green, that's actually the wavelength of light that's not being absorbed. Leaves appear green because chlorophyll reflects and transmits green light, but absorbs the other ranges of visible light. Therefore, as the other wavelengths of visible light are absorbed in the granum or of thylakoids, green light is transmitted because it is not absorbed, while the rest are used in photosynthesis to provide energy. A spectrophotometer, which you'll often use in your labs for general biology and general chemistry, measures a pigment's ability to absorb various wavelengths of light. This machine sends light through pigments and measures the fraction of light transmitted at each wavelength. Essentially, how much starting light is transmitted through a sample, how much of that initial light makes it through versus how much of it was absorbed in the sample. An absorption spectrum is a graph plotting pigment's light absorption versus wavelength. And here's just a general look. There's some light source that shoots a multiple variety of wavelengths of light that goes through a prism that selects, and then there's a filter that selects for specific wavelengths of light to pass through a sample. And some of that light does not make it through, and some of it does make it through the sample into a detector on the other end. And depending on that prism's orientation, we can get a range of different wavelengths. If we're just looking at the visible wavelengths, we can select for green in the top example or blue in the bottom example. And see how much is absorbed versus how much makes it through. What we see here is that as green light is shown through a chlorophyll solution, it's relatively able to pass through with high transmittance, which means 100% transmittance means it's all going through the sample and not interacting with what's inside. Whereas if we show blue light through the chlorophyll, it almost has zero transmittance or 100% absorption, because blue light can be absorbed by chlorophyll to power photosynthesis, whereas green light would not be absorbed and pass through. There are three types of pigment in chloroplasts. There's chlorophyll A, chlorophyll B, and carotenoids. Chlorophyll A is the key light capturing pigment, whereas chlorophyll B is an accessory pigment, and carotenoids are a separate group of accessory pigments. And effectively, these give you a range of scaffolding that, depending on the wavelength of light, one or more of these will be able to absorb energy of light and channel it down a pigment chain to where it can then be used. The absorption spectrum of chlorophyll A suggests that violet, blue, and red light work best for photosynthesis, and action spectrum profiles the relative effectiveness of different wavelengths of radiation in driving a process. Here we're looking at absorption with chlorophyll A. Seems to do phenomenal around the violet, blue spectrum of light. Chlorophyll B seems to do very well around more toward the blue end. Carotenoids seem to do very well in that region as well. And then further down, they seem to have certain various effects of orange and red light. So that the overall spectrum appears to do very well before or after the wavelengths for green and early yellow light. Anaerobic bacteria and filament of algae seem to have similar success. The action spectrum of photosynthesis was first demonstrated in 1883 by Theodor Engelmann. In his experiment, he exposed different segments of a filamentous algae to different wavelengths. Areas receiving wavelengths favorable to photosynthesis produced excess O2. He used the growth of anaerobic bacteria cultured along with algae as a measure of O2 production. The action spectrum for photosynthesis is broader than the absorption spectrum of chlorophyll. Accessory pigments such as chlorophyll B broaden the potential spectrum of light that can be absorbed for photosynthesis. The difference in absorption spectrum between chlorophyll A and B is due to a slight structural difference between the pigment molecules. Here we're looking at the difference between chlorophyll A and B. While largely the same molecule with a hydrocarbon tail that interact with hydrophobic regions of proteins inside thylakoid membranes, think of this as like an antenna that can absorb certain wavelengths of light and beam that energy or channel that energy downwards. The porphyrin ring is the specific end that's able to absorb light energy due to assistance due to the magnesium in the center. The only difference between chlorophyll A and B, however, structurally, is one little component that's either a methyl group in chlorophyll A or an aldehyde group, C double bond O with a C single bond H in chlorophyll B. Accessory pigments called carotenoids may broaden the spectrum of colors that drive photosynthesis, essentially allowing for more light to be absorbed. Some carotenoids function in photoprotein. They absorb excessive light that would damage chlorophyll or react with oxygen. When a pigment absorbs light, it goes from a ground state to an excited state, which is unstable. This means that it has more energy than it naturally wants to have. When excited electrons fall back to the ground state, excess energy is released as heat. In isolation, some pigments also emit light, an afterglow called fluorescence. This is not usually in the visible light spectrum, and sometimes it is. Excitation of isolated chlorophyll molecule. As a photon of light reaches it, it excites an electron, and as that electron lowers back into its ground state lower energy, that excess energy can be released as heat, as well as a new photon of light, which is the process of fluorescence, that afterglow after shutting off the lights. A photosystem consists of reaction center complex surrounded by light harvesting complexes. The reaction center complex is an association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor. The light harvesting complex consists of pigment molecules bound to proteins. Light harvesting complexes transfer the energy of photons to chlorophyll A molecules in the reaction center. These chlorophyll A molecules are special because they can transfer an excited electron to a different molecule. A primary electron acceptor in the reaction center accepts excited electrons and is then reduced as a result. Solar-powered transfer of an electron from a chlorophyll A molecule to the primary electron acceptor is the first step of light reactions. Here we're seeing a more in-depth version. On the right is the actual protein structure based off of its helixes and alpha sheets and other chains. But what we see here is a large complex absorbing light, which eventually raises an electron in energy level to a higher energy level. There are two types of photosystems in the thylakoid membrane. Photosystem 2, which was discovered later, functions as the first. The numbers reflect the order of discovery. The photosystem 2 complex is the first part of photosynthesis. This, however, was discovered after photosystem 1, hence the name irregularity. The reaction center chlorophyll A, PS2, is called P680 because it is best at absorbing a wavelength of 680 nanometer light. The second part of this photosynthetic setup is photosystem 1, which was discovered after photosystem 2. PS1 is best at absorbing a wavelength of light at 700 nanometers. The reaction center chlorophyll A of photosystem 1 is called P700. During the light reactions, there are two possible routes for electron flow, cyclic and linear. Linear electron flow, the primary pathway, involves both photosystems and produces ATP and NADPH using light energy. There are 8 steps in linear electron flow. First, a photon hits a pigment in a light harvesting complex of PS2, and its energy is passed along the pigment molecule until it excites P680. Now an excited electron from P680 is then transferred to a primary acceptor. We now call it P680+. And then we're in the electron transport chain, eventually forming ATP and taking another excited electron in photosystem 1 now to further down the electron transport chain. In the third step, water is split by enzymes, and the electrons are transferred from the hydrogen atoms of P680+, thus reducing it back to P680. P680+ is the strongest known biological oxidizing agent. The protons are released into thylakoid space, and dioxygen is released as a byproduct of this reaction. Each electron falls down an electron transport chain from the primary electron acceptor in PS2 all the way to PS1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. In the fifth step, potential energy stored in the proton gradient drives production of ATP by chemiosmosis. In PS1, as with PS2, transferred light energy excites P700, which loses an electron to the primary electron acceptor. P700+, which is now missing an electron, accepts an electron passed down from PS2 via the electron transport chain. In step 7, each electron falls down the electron transport chain from the primary electron acceptor in PS1 all the way to the protein ferrodoxin FD. NADPH+ reductase catalyzes the transfer of electrons to NADP+, reducing it to NADPH. The electrons of NADPH are available for the reactions of the Calvin cycle. This process also removes a proton from the stroma. And that would have been the final step. Now the energy changes of electrons during linear flow through the light reactions can be shown in a mechanical analogy, where essentially a proton first gives an electron excess energy, it loses some of that energy in photosystem 2, then another photon of light increases that electron again, and then it slowly loses energy in photosystem 1. In cyclic electron flow, electrons cycle back from ferronoxin to photosystem 1 reaction center via a plastocyanin molecule PC. Cyclic electron flow uses only photosystem 1 and produces ATP, but not the reduced NADPH. Also, no oxygen is released in cyclic electron flow. Here we're looking at a system where essentially the electron is cycled through multiple times without the production of NADPH. Instead, ferrodoxin takes it to cytochrome complex to produce ATP in that direction. Some organisms, such as purple sulfur bacteria, have photosystem 1, but not photosystem 2. Cyclic electron flow is thought to have evolved before linear electron flow. Cyclic electron flow may protect cells from light-induced damage. Chloroplasts and mitochondria generate ATP by chemiosmosis, but use different sources of energy. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transfer light energy into the chemical energy of ATP. Spatial organization of chemiosmosis differs between chloroplasts and mitochondria, but also shows similarities. In mitochondria, protons are pumped to the intermediate space and drive ATP synthesis as they diffuse back into mitochondrial matrix. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. Here we're just looking at both mitochondrion and a chloroplast side by side, comparing their structure on how they pump a proton, how they form a proton gradient that eventually goes through ATP synthesis. Getting pumped initially out of the matrix or the stroma through the inner membrane or the thylakoid membrane, and then into the intermembrane space or thylakoid space, then to pump it back through into the matrix of mitochondria or stroma of chloroplasts. The mitochondrion structure and chloroplast structure, again just looking at the transfer of protons, ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place. In summary, light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. And those are the light reactions. Now let's look at the Calvin cycle. The Calvin cycle uses chemical energy of ATP and NADPH to reduce carbon dioxide to sugar. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The Calvin cycle is anabolic. It builds sugar from small molecules by using ATP and reducing the power of electrons carried by NADPH. Carbon enters the cycle as CO2 and leaves as sugar named glyceraldehyde 3-phosphate, or G3P. For net synthesis of 1G3P, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide. The Calvin cycle has 3 phases. First, the carbon fixation, catalyzed by Rubisco, then the reduction phase, and lastly, regeneration of carbon dioxide, and the acceptor, Rubisco-P. Here we're looking at the start. as 3 molecules of carbon dioxide are coming in, reacting to form 3-phosphoglutarate, then 1,3-bisphosphoglycerate, and then G3P, which can then be used to output G3P, and then glucose and other organic compounds from there. But in the cycle, G3P can cause a regeneration of rubisco-P, rubiscophosphate, which has the process of using 3 ATPs to reestablish rubisco P, and then becomes rubisco. Alternative mechanisms of carbon fixation have evolved in hot, arid climates. Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close the stroma, which conserves water, but also limits photosynthesis, as carbon dioxide cannot go through a closed stroma as easily, and it results in a buildup of O2 inside the stroma. These conditions favor an apparently wasteful process called photorespiration. In most plants, referred to as C3 plants, the initial fixation of CO2 via rubisco forms a 3-carbon compound, 3-phosphoglycerate. In photorespiration, rubisco adds O2 instead of CO2 in the Calvin cycle, producing a 2-carbon compound. Photorespiration consumes O2 and organic fuel and releases CO2 without production of ATP or sugar. Photorespiration may be an evolutionary relic because rubisco first evolved at a time when the atmosphere had far less O2 and more CO2. Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. C4 plants minimize the cost of photorespiration by incorporating CO2 into 4-carbon compounds. There are 2 distinct types of cells in the leaves of C4 plants. Bundle sheath cells are arranged in tightly packed sheaths around the veins of the leaves. Mesophyll cells are loosely packed between the bundle sheath and the leaf surface. Sugar production in C4 plants occurs in a 3-step process. First step is the production of the 4-carbon precursors is catalyzed by the enzyme PEP carboxylase in the mesophyll cells. PEP carboxylase enzyme has a higher affinity for CO2 than rubisco does. It can fix carbon dioxide even when carbon dioxide concentrations are very low. Step 2, these 4-carbon compounds are exported to bundle sheath cells. And in step 3, within the bundle sheath cells, they release carbon dioxide that is then used in the Calvin cycle. Comparison of C4 leaf anatomy and the C4 pathway that was just discussed. Since the Industrial Revolution in the 1800s, carbon dioxide levels have risen greatly. Increasing levels of CO2 may affect C3 and C4 plants differently, perhaps changing the relative abundance of these species. The effects of such changes are unpredictable and a cause for concern. Suitable agricultural land is decreasing due to the effects of climate change, while the world demand for food continues to increase. C4 photosynthesis uses less water and resources than C3 photosynthesis. Scientists have genetically modified rice, a C3 plant, to carry out C4 photosynthesis. They estimate 30 to 50% increase in yield compared to C3 rice. Some plants, including succulents, use cressulicin acid metabolism, or CAM, to fix carbon. CAM plants open their stromata at night, incorporating CO2 into organic acids that are stored in the vacuoles. Stomata close during the day, and the stored CO2 is released from organic acids and used in the Calvin cycle, while light is also available. The CAM pathway is similar to the C4 pathway in that they both incorporate CO2 into organic intermediates before it enters the Calvin cycle. The C4 pathway structurally separates the initial steps of carbon fixation from the Calvin cycle. In the CAM pathway, these steps occur in the same cell, but are separated in time of day. For CAM pathway, essentially CO2 becomes an organic acid while at night, and then during the day, CO2 is regenerated and goes into the Calvin cycle while light energy is available. Thus, temporal separation of steps, but in the same cells. Whereas in C4, CO2 is incorporated into organic acids into one type of cell, the mesophyll, and then in a separate cell, carbon dioxide is released into the Calvin cycle for the bundle sheath cells, thus spatial separation of steps. Sugarcane and pineapple being an example of C4 and CAM plants respectively. Light depends on photosynthesis. The importance of photosynthesis, a review. The energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. Sugar made in the chloroplast supplies chemical energy and carbon skeletons to synthesize the organic molecules of cells. Plants store excess sugar as starch in chloroplasts and other structures, such as roots, tubers, seeds, and fruits. Just looking at the full picture of what goes on in chloroplasts, from light reactions to Calvin cycle. And then a more in-depth look of the transport of various molecules all across a plant cell to form a functioning unit. That is insane. Apparently cell membrane movement is more relevant to chapter 7, which is not usually touched upon too intensely in general biology. All right. We covered a long way. Corn plant surrounded by invasive velvet leaf plants. In this case, if you are interested, subscribe, like the video if you've made it this far, just because you've heard it time and time again. But I'm a small channel, and really, to be honest, if YouTube ends up paying me someday, that'd be cool, and you guys and gals could be a part of helping that. And if not, totally understandable. This isn't my main profession. In any case, this concludes our look into photosynthesis for this chapter of Campbell biology. So just past the 39-minute mark, wishing you all well, and in all your classes, peace.